This one only has the innermost layer. So if I stick a sticker on the outside of a kidney model, that's the fibrous capsule. You know, we'll talk about other like fat capsules like later in lecture. But you can see if I stuck it on the outside of the kidney, that's the fibrous capsule. The kidney has two major regions. You have the cortex, which is on the outside, and the medulla is on the inside. So if I want you to have cortex or medulla as an answer, I'm asking region in the question. You know, we deliver the blood to the cortex. You know, and that's why you can see a lot more blood shown here. So cortex, outside, medulla, inside. Let's see, like really some books will say three regions, where it's like they'll consider the renal pelvis like a region. It's more like a reservoir, a P reservoir, but renal pelvis, but I'll point to that again probably. So when the medulla, some of the main things are the pyramids, the renal pyramids. In between the pyramids, you have renal columns. So the tissue in between each pyramid is a renal column. So pyramid looks like a triangle, and then the space in between them are the columns. Most likely I wouldn't ask column. You know, I would, if I, you know, a column is probably one that's more covered just for completeness. The very tip of a pyramid is called a papilla. The tip of the pyramid is called a papilla. And they'll actually talk about papillary duct. I don't stress papillary duct in lab. I'm pretty sure I don't say have it written anywhere on there. So, but the tip of the pyramid is a papilla. And then the renal lobe says one pyramid in the adjacent cortex. And so this is a pyramid, and then the part above it, cortex. So this is kind of like one renal lobe here. It's not really a great representation because it's not showing it being like triangle shaped for the pyramid, but still it's like cortex, medulla. So one lobe is the medulla and the associated cortex that's adjacent to it. So this model is saying, well, let's look at one lobe. And then it's saying, well, let's look at one of these little red balls over here. It's just zooming in at each level. Mm-hmm. So we get to the calyces, minor calyx, major calyx. They're little pea cups. I'm a little pea cup. And so the papilla goes into the minor calyx. And then multiple minor calyces fuse to make a major calyx. So it's like these minor calyces, and like this is probably the best place where it's ripped open, where it shows where like a major calyx would be. So it's probably the best place to have a major, but two little things fuse to be a major calyx. And then major calyces fuse to make the pelvis, the renal pelvis. So the pelvis is like a big lake, a big reservoir full of pee. And then it's fed by tributaries called calyces. I've never used the word tributary ever before, maybe, in anything. So let's see. Renal pelvis, I just did. Renal sinus, it's more yellowish on this model, where the renal sinus is just the space that the renal pelvis is sitting in, and it's like that adipose in it. So the renal sinus is just, like the kidney like indents in, it's just like the cavity where the renal pelvis is located. So if I was asking a renal sinus, I kind of like it on this model more because it's obvious, it's a much different color. Like here, they're putting the adipose tissue the same color as the calyces in the pelvis. So I like how on this one, if I did do the sinus, I would do it on this one where it's bright yellow and it's obviously different. Okay, so nephrons are the units that do the work in the kidney of making pee. So you can't see nephrons, well, you kind of can here. You can see nephrons here, but zoom in, and then here are the nephrons on this. And so there's two types of nephrons. We have a cortical nephron and a juxtamedullary nephron. So the cortical goes way down deeper into the cortex. So this model has two different cortical nephrons. And then the one that's more, hold on, I just said this backwards. Said it backwards. I hate you guys are recording when I say something wrong. Cortex is on the outside. We have one cortical nephron, two juxtamedullary nephrons. Ugh. So the one that's mainly in the cortex is cortical. So 2 juxtamedullary, 1 cortical for the third time. 2 juxtamedullary, 1 cortical. Because likely one of your questions on your lab exam will be, what type of nephron is this? Like I guarantee I'll ask one question. So keep the one that's mainly in the cortex, which is the outside, just like the outside of your brain is called the cortex, is cortical.
Wait, is this the cortical or is this the cortical?
This is not, this is something different.
Okay.
This.
Okay.
Okay, so nephrons, how it says it has 2 parts. You have a renal corpuscle, which is the red ball, and then you have the tubule. So we talk a lot about this in lecture, where we say in the renal corpuscle, I'm going to make filtrate. It's where I take my blood and I basically just force stuff through a capillary, through a filtration membrane. I force stuff through, and then as it passes through this tubule, it becomes urine. So that's the 2 parts of a nephron, are the renal corpuscle and the renal tubule. So on the renal corpuscle, it has a couple structures listed to know about it. It says the glomerulus, and then it says afferent and efferent arteriole. So the glomerulus is a capillary inside of the renal corpuscle. Glomerulus means little ball, and we talked about that last semester when I talked about a glomerulus in the olfactory system. So little glomerulus, fenestrated capillaries. It's lab. I'm not asking about it being fenestrated, but it's got the little pores in it. The little windows that are going to let stuff go through it relatively easier. And then how I get in, afferent. How I get out, efferent. So the afferent arteriole goes into the capillary, the efferent arteriole goes out. So arteriole in, arteriole out. On this one versus this one, afferent in, efferent out. And notice how the afferent's a lot bigger. The door going in is bigger than the exit, so then you build up a lot of pressure and it makes you force the stuff out to make filtrate.
So would you probably label it on this one then?
I could do it on either, because you just know the door in versus door out. And so afferent in, efferent out, because I'm pushing through this filtration membrane, through this capillary and through these cells, and I'm pushing out filtrate to fill up this little ball. And then when it goes down and it passes through the tubule, we turn it into pee, we turn it into urine. So glomerulus is one part of renal corpuscle. And then the other part is called Bowman's capsule. So Bowman's capsule is almost like synonymous with the renal corpuscle. So like Bowman's capsule is basically the red ball part. It's not the capillary, it's the outer bit. So how it says you have a parietal layer and a visceral layer. Remember, viscera means organ, parietal means cavity. So this is a cavity in between the capillary and the outside. So the outside of the renal corpuscle is the parietal layer of Bowman's capsule. I don't really care about you saying, you won't probably have to say in lecture, you might have to use parietal. Lab, I don't see you having to use parietal or visceral layer, but it's something you have to understand in lecture. So, because it's 2 different layers, parietal layer, and then these cells, that's what you will likely have to say in lab. The green on this model, these pink on this model, that's the visceral layer of Bowman's capsule. But they're made up of cells called podocytes, which means foot cells. And so you likely, I know you will not have to say parietal layer or visceral layer on the lab exam. But I listed on our lab sheet because it helps us to also talk about lecture. You have to worry about this inner layer of Bowman's capsule is called podocytes. The cells are called podocytes. And you can ask me questions as we go on here. Then we get to the renal tubule. And there's regions. You won't just be saying renal tubule as an answer. It's kind of like like the colon. You have to say the part of the renal tubule. And so when I first come out, it's yellow on both models. When I first come out of the corpuscle, and the fluid first is going in, it's called the proximal convoluted tubule. Proximal, closer to point of attachment. Convoluted, because it zigzags back and forth like a crazy straw. Proximal convoluted tubule. And then I go down the descending loop of Henle, and I come up the ascending limb of the loop of Henle. So I'm pretty sure all you really have to say is loop of Henle rather than ascending or descending limb for lab. You need to understand the limbs more for lecture, but that's the loop of Henle. And then when you get up and it zigzags back and forth, and now it's tan on both models, that's the distal convoluted tubule. Distal means further from point of attachment. So once again, proximal convoluted tubule, loop of Henle, distal convoluted tubule. Now I've made urine, and then it delivers to this structure that goes down right here. That's called the collecting duct. And the collecting duct concentrates your urine. I'm not asking functions in lab, but I'm saying it concentrates your urine as we go down towards this. Collecting duct's gonna lead to the papillae, and then take your urine to the minor calyx, major calyx, and then it's going to go, why do I leave my kidney through what did you just say? Your ureter or ureter?
Ureter. But that's right.
Yeah, but I think you said ureter.
I don't know how to say it.
It's okay.
Ureter or ureter. So that's, if I want it ureter, I'd do it here. If I'm up here, it's the pelvis. So I would be down if I went, I like to call it ureter, just to really, even though that's not how it's pronounced, but just because it exaggerates it. But you said UT first. You said uterer.
Whatever.
So the juxtaglomerular apparatus, big lecture concept, also something that likely will get asked in lab, like maybe likely. And so this right here would be going to the proximal convoluted tubule. Notice how the distal convoluted tubule, when it comes up, it kind of folds back and goes by the Bowman's capsule. So this right here is the distal convoluted tubule on this big zoomed in part. And this is the afferent arterial. So if I put a piece of tape on both of them and said, what is this? That's the juxtaglomerular apparatus. So we talk about that in lecture, about what it does and what this part regulates and what this part regulates. But it's regulating the formation of filtrate, what we call our glomerular filtration rate. And so these two things work together, the afferent arterial and the distal convoluted tubule. If I put a sticker on both of them, and it's easier to do on here than it is on this one. It's hard to put a sticker on both of them on this one. So if I did juxtaglomerular apparatus, when I do juxtaglomerular apparatus, it would be on this one. And we talk about this in lecture about how it regulates the kidney. And so now we have to talk about blood flow, like blood flow in the kidney.
Please.
And you have to know blood flow for the lecture and for the lab, like this thing about blood flow. And it's pretty much listed in order. Like the blood flow is pretty much listed in order. You know, renal artery, you come in. And then that first branch off the renal artery is going to be segmental artery. You know, first you go to segments, and I'm saying it's listed in order. And then you're in between the lobes. Remember we say the lobe is one pyramid in the cortex. So in between the lobe, interlobar. And then it says arcuate. Arcuate is like an angle. Think of like the arc of a jump shot or something. That is arcuate artery. So arcuate. The next thing it says is cortical radiate. Some books will call it interlobular. I don't like interlobular because you confuse it with interlobar. But it's radiating out into the cortex. So these ones that are coming off arcuate going out, they radiate, so cortical radiate artery. And then we get to, you know, off of that, you can see it basically jump in to afferent and efferent arterial. So they're kind of showing the afferent arterial branch off of it, but we have to like zoom in to see afferent and efferent arterial. We don't have to worry about peritubular capillaries or vasa recta being asked on lab. We'll worry about those in lecture. Peritubular and vasa recta, those are going to be associated with the tubule, and you're going to see what we're talking about when we get to some pretty important concepts in lecture. But you won't be you'll just have afferent, efferent arterial, you know, afferent, glomerulus, efferent, and then it goes to these structures that are capillaries, but we're not going to worry about those in lab. They're too hard to ask on the model. And then the veins, it's just opposite. Cortical radiate vein, arcuate vein, interlobar vein. And you don't have a segmental vein. It just goes from interlobar to renal. There's no segmental vein. And seen.